Excellent, excellent. So, the aging is a big trouble and a risk factor for everything. Here are only 4 diseases, but in reality, there are much more and more. So if you look at the NIH budget prior to the trouble, kind of a substantial amount of money was allocated to solve aging, still less than infectious diseases, still less than cancer. But if we look at the distribution of this money, we will realize that most of it goes to Alzheimer. So that the fundamental aging research is funded much more modest, like it's less than 1% of entire NIH budget, which is sort of disproportional to the scale of a problem. Moreover, some of the heads, like leading minds in the field and that affect the organizational side as well, they are advocating a switch from lifespan extension to health span, meaning that we don't need to target this key common cause for all these troubles, but we need to target each of the curves individually, which is a very strange take, right? Because it seems to be if there is a single cause, then there should be a single drug that will remove all these curves. Why does it happen? Additionally, we have some drugs that may slow aging, rapamycin, metformin, Ozempic, all were discovered by chance. The gerontologists have little to no idea why they work. And so these drugs were discovered not by gerontologists. And it seems to be that with or without gerontologists, the progress in lifespan extension will go roughly the same speed. So what is the bottleneck? I believe the main bottleneck is that we don't know what is aging. So this is a paper from 2024. Vadim Gladyshev was the lead. So they just asked different experts in the field what is aging, got completely different answers, and got this word clouds. So finally, what is the most important unanswered question in the field? So the big hit is aging, right? So that we have no scientific paradigm for it. And why do we need scientific paradigm? Because we must make choices based on scientific paradigm. And the very first fork that we need to pass is aging damage accumulation stochastic entropic, or it's a program that kills us. These are two very different models. In the first case, we need to make these dots go their places back or make them fly away slowly. In the second case, we need to find this gun and remove the bullets. And these are two absolutely different projects. In one case, we need to repair something. In another case, we need to break something down. If we pass this fork in the wrong way, we lose all the time and resources invested. And this is only the first fork. So there will be more forks later, of course. So what are the approaches to answer the question, what is aging? So the main approach so far is biochemical. So that people are suggesting different aspects, mechanisms, biochemical pathways, molecules that can be key for aging. There are like 300 hypotheses proposed around. They are not contradictory. So that the problem of biochemistry is that it's incredibly complex. And if I suggest that aging is caused by some molecule here, the other person comes with an experiment that doesn't fit the model. And why the ends do not meet the ends, I can always hypothesize that something in this corner affects aging, and this brings things together. And it's incredibly difficult to disprove the models. As you may know, this oxidative damage model from Harman got like a couple decades to disprove. That's why I believe the right approach to the problem is to look at ecology and evolution and try to explain the phenotypes that we see in nature to approach to the real cause of aging. So you know this quote that I don't need to repeat it probably. If we see at animals, we find incredibly strange phenotypes, aging related. Some animals do not barely age. Some die after reproduction. In some cases, one genome, like in eusocial insect, can give two very different lifespans. So like queens can live 50 times longer than workers. Whatever is flying live very long, and there is no good model to explain that. So far, there are only 2 to 4 evolutionary hypotheses of aging, and this is much smaller cohort to deal with. So the key question that I want to address is why different species are living so differently. So rat lives 4 years. The squirrel can survive to 20. Same size animals, relatives, same diet. What is the difference? So the traditional take is that the efficiency of damage control is adjusted to a particular ecological niche, and people often cite extrinsic mortality, maturation time, and metabolic rates as something given and something very well studied, boring, and simple. In reality, it is not, because if you look at animals from different taxa, mouse size, and very long-lived at the same time, like this is Brandt's bat, a sparrow, or a frog, we find that they all mature very fast. These two have very high metabolic rates, and these two have high extrinsic mortality. So all these factors that are classically cited, they have clear exceptions that are not explained very well. This rat and squirrel is only part of the spectrum. On long-lived side, we have this naked mole rat that doesn't age at all, so it can survive to 30 to 40 years without increase in mortality. On the other side of the spectrum, we find similparous mammals. This is northern quoll, an animal that dies after reproduction. This is specific only to males. So after mating, after the breeding season, all the males die because of some hormonal effect. And the question is, where is damage in this equation? So does entropy affect rats differently from squirrels? So of course, we see damages in aged organisms, for sure. But we also see damages in many, many other diseases. And we don't suggest damage because of COVID. Well, if you take a COVID patient, we can find a lot of damage there. And this is a question of the language we use to approach approach aging, and the question of what are our input observations. And with this, I'm very different from everybody in the field, because most of the people come from the side of chemistry to find the chemical modifications to the molecules that damage them, or like physics and entropy, or you are coming from this morphostatic collective communication perspective. So these all are different languages, but from my point of view, chemistry, physics, or collective behavior are coming from the same side, because they are striving to explain what happens in the organism. I'm striving to explain why do organisms evolve to reach a certain pace of aging. So, like coming back to evolutionary points, so I use evolutionary methods. So there are 3 classic evolutionary theories: selection, shadow, antagonistic pleiotropy, disposable soma. I will rapidly update you on this. Do I need to do that?
Can you just say what selection shadow is? I'm not familiar with that one.
So this is the first idea, was that actually animals do not survive to the age of aging in the wild. So, and if nobody survived to this point, then it doesn't matter what happens. So, but it is empirically wrong, because if you take lions, let's say wild population, we see that the mortality increases with age. A lion of 14 years has higher probability to die per year than lion of 5 years, meaning that lions age in the wild.
Yep.
So that this is not sufficient to explain aging. And so the next one was G.C. Williams, who tried to complement this selection shadow, his idea of pleiotropic genes, that the genes are good in youth, bad in old age. And if we accept the existence of such genes, this is okay, because the beneficial trait is good for many animals in here, and detrimental affects only this, and the net benefit is still very positive. But the problem is that this requires this very evolutionary rigid pleiotropic genes, and it's not clear why evolution cannot disconnect or compensate the detrimental trait of the pleiotropic gene. So Kirkwood, disposable soma, suggested the existence of the fundamental trade-off between reproduction and longevity. We have limited amount of energy. We can invest it either in reproduction, either in longevity. If we overinvest in reproduction, we can just over-replicate the slow reproducing but long-living strain. So this was an important breakthrough, like intellectual, because it suggested a fundamental trade-off between aging and something, but energy is likely wrong currency. The model has mathematical problems, and it has many biological problems, like why caloric restriction prolongs lifespan. So we remove energy, why lifespan increases. Males invest less in reproduction, live shorter than many mammalian species. Food consumption decreases with age, should be other way around. If we can take more food and fix ourselves, then we should do it, probably, but we don't. Also, all the poor ecosystems where animals should save, they are full of centenarian species, like in Arctics and caves in this deep water. And this is not clear why, how to fit all these connections. And from the evolutionary point of view, all the damage accumulation tropic models are based on these two questionable theories. But why people, and these theories are quite bad in explaining variability, association with flight, why different castes of social insects live so long, what the heck with naked mole rats, similparity. So all that is very poorly explained by these models. And I want to emphasize that damage accumulation models are not proved experimentally. So we don't know about molecules that accumulate damage and drive aging. Still, it is a hypothesis, a theoretical one. But why people stick so strongly to the damage? Because we historically have had two very strong arguments against program. So this choice is made not because there are good reasons to believe in damage, but because there are good reasons not to believe in program. So these arguments are what the program good for. And why don't we see immortal mutants in our experiments or in humans or in nature? So, and I have built a model that can address both these questions. So first of all, what the program is good for. In principle, it's known the adaptive suicide in bacteria or apoptosis in unicellular organisms, if infected with viruses, they try to kill themselves to save their neighbors from the infection. So they will die anyway, but they want to also kill the virus together with them, that it doesn't infect relatives. So this is very well characterized. Viruses are trying to overcome it. Bacteria is trying to overcome virus inhibition, invent other stuff to kill themselves. So this is very well accepted part of the immune system. So can something like that happen in metazoans, in higher animals? Let's say we have a chronic infection, like this parasite in this fish. Let's say probability is constant per year. Let's say then the older the fish is, higher the probability is that it is infected. And at certain age, let's say after age of 8 years, every fish will be infected. This particular parasite doesn't sterilize much, but let's say the parasite doesn't let the fish to reproduce. then this older fish is useless for its inclusive fitness. It cannot reproduce itself, but it can infect its relatives that swim nearby. So, and the key assumption of the model is the population viscosity, that if an animal infects its relatives with higher probability than non-relatives, then aging can exist to remove this heavily infected individuals. So, and to model these viscous populations, because there are not many mathematical models developed before. So we model territorial animals that move like charged balls on the surface of a dielectric, and you see trajectories are similar to this territorial movement of wolves, GPS tracked in Minnesota. So this model recapitulates two very important properties of the population that can be first universal, or more or less universal. Second, they are typically overlooked in the classic epidemiological models. So first of all, this is density-dependent dispersal. So animals from densely populated areas are migrating into sparsely populated areas. And second, these kinship gradients, that animals of related animals are interacting and infecting each other more frequent than non-relatives. So, and let's see how the this model works. So these are yellowish are short-lived, reddish are long-lived. So the black rings are infections. You see that infection is much more prominent in long-lived strain, because just animals spend more time in the infected state after age X. So at the same time, if in the absence of the pathogen, we compete these two, too early. We compete these two strains, then long life wins, because they just live longer, live more progeny, and take over very, very rapidly. However, in the presence of the pathogen, situation changes to the opposite. So you see that first these long livers are expanding, but then the pathogen comes into the play, and they are preferentially infected because they don't clear these infected older ones, and they can be displaced by short-lived strain, which has this non-infected patches that produce like population pressures. So important to emphasize, this pathogen is sterilizing. So it doesn't kill, doesn't shorten lifespan, but it doesn't let animals to reproduce. So in our population, these are probably sexually transmitted pathogens like gonorrhea, syphilis, or maybe HIV also has some effects on fertility. So with this, we answer the first question: what is the shorter lifespan or aging good for? It is to remove chronically infected individuals. Second question we want to answer is, why don't we see mortal mutants? Indeed, we see something that we can believe is a gain of function mutant, a progeria, right? But we never see Voldemort that is an immortal mutant, and we can treat our flies with the mutagens, and we find flies without eyes, without wings, but we never see ageless fly. So, which is sort of counterintuitive. So the aging should be something more important than flight or vision. Strange, right? However, we know that immortal mutant can exist, because we know that naked mole rat doesn't age much, but it share a common ancestor with other rodents. So somewhere along this branch, a mortal mother should have produced an immortal baby. It is possible technically, but it's clearly very, very rare. So by some reason, aging is very well fixed in our genome, that it is not easy to mutate in one go. Questions? No? Okay, probably it was eco. Let's look at a species where aging is not fixed that well. So these are eusocial insects, ants, where, as I told, queens are living like 50 times longer than workers. So this paper showed that some pathogens are coming in and prolong lifespan of workers to make them as long-lived as queens. So, and this is clearly understandable from the point of the pathogen, right? The pathogen wants us to live longer, to spread longer, and to improve its dissemination. So, idea is that organisms do not want, evolution doesn't want pathogens to manipulate our aging. And and that's why it evolved some security harnesses that prevent aging from manipulation by pathogens, also by mutations, and also by therapies. So this phenotype is not limited to this paper. In principle, you can also find something like that in humans. It was shown that some nematodes can secrete immunomodulators that can reduce mortalities also in humans. So idea is that I need to say this is not the only argument that pathogen control hypothesis produces to explain absence of immortal mutant, but I don't want to go to another one because it's more like population dynamics heavy. So with this we answer both arguments. We know what's aging good for. We know why we don't see the mutants, and I think there are no any more reasons to believe in entropy and damage. We need to focus on the program. So this hallmarks of aging diagram, I believe chronic inflammation is the key thing there. And I need to say that if we compare different observations made in nature, then pathogen control explains them better than any other model. So unfortunately, I don't have time to go through all the 25 observations and discuss it from the point of each theory. You can go to my this paper, and I will be happy to argument if you disagree. I want to now focus only on two craziest predictions that we have and that we want to test experimentally. So first of all, question number one: why do naked mole rats live so long and don't age? So the classic model doesn't have any explanation for this. So our ecological explanation that they have redeveloped this adaptive killing in response to infection. This is a harakiri knife. So in principle, there is alternative way how to control pathogens. If the very first animal that got infection kills itself immediately, then the colony can be kept free of pathogens. So then aging can become redundant because there are no pathogens anymore, so we can model it. The problem is that in mammals, we have tons of other like light pathogens like flu or common cold, and killing self in response to all that would be a little bit wasteful. And the immune system likely cannot discriminate between HIV and flu. So, and that's why, like in most species, this type of response, this, blue one is this strain that is long-lived and also has this phenotypic response, kills itself in response to infection. So idea is that the flu would kill this strain very rapidly. But eusocial structure of naked mole rats allows this response. And if you look, yes, please.
Just one question. So would you also predict that animals that are more socially isolated would then live longer because they don't have to deal with this long lifespan issue of like transmitting diseases?
This is a deep question. So in principle, yes and no. So because I think in mammals, most of the pathogens of interest are sexually transmitted. So, and it doesn't depend really how solitary the animal is. So because it still needs to reproduce, and there are like frequency and density dependent transmissions, and sexually transmitted pathogens are frequency dependent. So they don't affect it, and there can be opposite effects because if animals are infecting non-kin individuals more often than kin, then the model is completely screwed up and long life should be favored. So, and that's why if animals have like bats, they have huge communities by like thousands animals and promiscuous interactions, promiscuous infections likely, then it should promote longevity. So, in brief answer to your question, pathogen transmission network can be very different, and for solitary and for social animals. So it should be considered each species by species. So, yeah.
Sorry to interrupt, just to follow up on that. And we know that these sexually transmitted diseases are linked with lower lifespans, like gonorrhea or whatever does affect the lifespan. I guess that part isn't clear. How do we make this link between these pathogens? Like, it's just about sterility is what you're talking about, right? So these STIs sterilize the animals, and so they are unable to pass on their genes, and so as a result, the population loses those long-lived animals. Is that correct?
No. Aging is part of the immunity. It's not caused by a disease. Aging is part of the immunity that prophylactically kills animals by the expectation. So when we expect animal to get a disease, by that time it should die. So it is immunity. It's not a consequence of the infection. Sorry if I wasn't.
Okay, no. Okay, that makes sense. So, okay, cool.
Yeah. So then there are a couple papers that indicate that sort of point towards that innate mole rats are indeed like suicidal. So in this paper, they tried to introduce gene of interest into innate mole rats using herpes simplex virus as a vector. And all the mole rats died. Very sad case report. Precious animals don't do that. And here they have also coronavirus natural infection, but this shows rather that queens are less susceptible to dying than workers, which is also one of the predictions of the model. But still, this type of models are still in progress, so we haven't done them yet. So idea for this negligible aging evolution in innate mole rat is that key was satiality, then they developed suicide in response to infection, and after that they got negligible aging. So another crazy project is about similparity. This is similparity is dying after reproduction. So the classic example is Pacific salmon that goes up the river, lays eggs and dies. So but we are interested in evolution of similparity in carnivorous marsupials. In this clade, it evolved like 5 times independently. And in all the other mammals, never, which is very, very strange. So, and there is no explanation for that. So we believe this should be like pathogen control for very specific type of diseases. And indeed, the largest animal in this clade has a very unusual disease. This animal is not Similparus, it's Itheraparus still. This is Tasmanian devil, and you know that they have these tumors, transmissible tumors, that the cells of the devil can be transmitted from devil to devil by bites. And this disease has very unusual properties epidemiologically, that they are this disease is very transmissible, very lethal, and has a very long incubation period. There are not many such diseases in the in nature, and importantly, the disease peaks during the breeding season. So idea is that during the breeding season, the devils bite each other or other marsupials, transmit this tumor, then the probability of surviving to the next breeding season is negligible, and this infected animal will just go around and spread disease between the breeding seasons by fighting sometimes occasionally for food with its relatives. So it is better to kill these infected males prophylactically before, immediately after the breeding season. And we can model it, and indeed, without disease, iter parity wins, with disease, similar parity, but wins, but this is still in progress, as Yuri is trying to, this was started by a genius student working with me at UCSF, but now we are like finishing this project with Yuri. So the idea, the key prediction would be that we predict prevalence of transmissible cancers in other marsupials. Ecologists do not believe it, and I believe that if we will find evidence, then it will be a strong prediction that people wouldn't be able to ignore. And this is also important to mention that DFTD, this devil's facial tumor disease, is present as two strains. So one strain evolved like 30 years ago, another one 20 years ago. So the devils are definitely very susceptible to induce this disease de novo. And this is also sort of argument in our favor. So this is a physiological model why marsupials can have this predisposition for transmissible cancers, that they have they don't have placental suppression of the immune system, but the embryo is attaching to the wall of uterus, and this is accompanied by inflammation. And so they can be like in a trade-off between non-self-recognition to prolong the attachment period and better nurture of the embryo, and and non-self recognition. And there are like small case reports, again, supporting this point of view that they don't have some types of non-self recognition, but all that need to be tested. So idea is that this embryological property leads to poor non-self recognition, leads to transmissible tumors, leads to evolution of similar parity. So still very early stage, hypothetical. So these are two ecological hypotheses we want to test. So why doing crazy projects? This is my family doctor who influenced me a lot. So he always was saying that first we put the diagnosis, then the treatment. So I believe that before trying to cure aging, we need to find what it is. And for that, we need to do crazy stuff sometimes. But on top of that, we have a more conventional project in the lab. Marina is now working on this. So according to the pathogen control model, aging is part of the immune system. But what about cellular senescence? Cellular senescence, you know, it's another hallmark. And we believe it's also part of immunity. I don't need to introduce you to cellular senescence, right? You know this, yeah. So, but this, the left panel is a screenshot from the site from this ICSA. I'm also a member. So, International Cell Senescence Association. And this is a definition. What is cellular senescence? Cellular senescence is a programmed state of stable cell cycle arrest that is accompanied by a complex phenotype. Okay, this is an observation. Next. Senescent cells play a role in physiological processes such as tumor suppression, wound healing, embryonic development, whilst paradoxically they can contribute to aging, cancer, age-related disease. Now, this is much less clear, right? So, you know, cancer is present for two times because, you know, the initially senescent cells were considered anti-cancer, but then the people realized they are pro-cancerous. And so the induction of senescent cells and their rapid killing seem to be beneficial in many systems, but accumulation of senescent cells was shown to be bad in everywhere. And what for this evolved? Because it is a complex phenotype. It's like orthogonal cell biology. Super interesting. We are very excited about this project. But what for are they needed? So this is not clear. And it was a paper published like almost 10 years ago that cell senescence is antiviral defense mechanism. Unfortunately, this paper has like minimal flaws. So some things were not convincing enough and was largely ignored. So, but, you know, we applied minimal effort, we reproduced the results, extended them a little bit. So these are control cells infected with a GFP labeled virus. And these are senescent cells, and you see the difference. So the senescent cells are clearly antiviral. So I don't want to go into mechanistic detail, but if we inhibit some of the JAK-STAT pathway, the immune pathway, we can rescue the virus. So it is about immune system. It's not about like they are weak and sick and cannot support virus infection. So
just a question on this. What happens if you put you co-culture senescent cells with non-senescent cells? Does it extend the protection?
Yes, okay. Yes. They extend protection to they do interference basically. So the interference type 1, so the healthy cells that sit next to the senescent cells are protected. And our big model is this immune militia model hypothesis of cellular senescence, that in response to infection, healthy cell can become senescent. So recruited from any other job to this, duty, work on defense against viruses, thus protects the neighboring cells. Also, if we just take conditioned media from senescent cells, put on the healthy cells, they are also protected. So the SASP likely communicate with the immune cells to help them to manage the situation. Then, after the war is gone, the senescent cells must be demobilized because they can still contain virus. So it can be damaging if they go back to their original job. So, and if they accumulate, if this is broken, then they start looting. So this can be this can be like indication that the infection is still going on, or it can be a part of the adaptive aging program. So, and so far, most of these arrows are supported either by our experiments, either by other people's data. So I think this is the most consistent model for cellular senescence proposed so far. So and just as a potential opening to the brainstorming, what can be like interaction between our labs. So that some animals can switch aging on and off, and this Hydra and also Planaria, and paradoxically, they can do it at low temperature. They do it at low temperature that prolongs lifespan in many other ectotherms, which is a little bit, also challenging the classic dogmas. Also, I heard that Planaria do it in high population density, and it also associated with sexual reproduction in both, I think, in both cases, right? So as far as I understood, the aging in Planaria itself has not been shown, but telomere shortening has been shown, right? So, and the potential hypothesis is that switching on the aging as a pathogen control mechanism, so that in presence of pathogens, aging can be switched on. And then the hypothesis number 2, a little bit off, that also sex can be a pathogen control mechanism. We haven't modeled it carefully, but I think we can find gonorrhoeism or sexual reproduction, at least in our models as well. Because you know that evolution of sex is also a big paradox. So because specifically we need a little bit of sex, like 10% of reproductive events may require to ensure a normal level of recombination. But the gonorrhoeism, I think, is not explained at all, because males are basically useless. And I think we may find it in our models of this pathogen control in viscous population. So this probably a teaser for a discussion. And these are people who work with me at UCSF. These are people who work with me right now. And I'm happy to take questions. And yes, thank you to the funders.
Thank you so much. That was great. Questions?
So just to follow up, What about species that don't have sexual reproduction, like certain lizards, for example, that clone themselves through like mimicking sex, where the daughter or the mother like gets on top of the other one, and then basically it just like produces eggs that they fertilized. Do we see like longer lifespan in those type of animals where they kind of mimic sexual reproduction but don't have sex?
No, so I never looked at this, frankly. I don't know. So I think it all depends on the specific pathogens and because I'm not sure. Probably in reptilians also sexually transmitted pathogens are of interest. Because, for example, in so the pathogen should be chronic and sterilizing. Most of these in human population are sexually transmitted, right? But in, let's say, in invertebrates, no. In invertebrates, like cestodes and fish, they are not sexually transmitted. So it also is depending on the taxa. So I don't know about the lizards, frankly. And I think these lizards are probably not a very long, do they have a long evolutionary history? Because I was taught that this loss of sex goes into the decline. So
It's a good question.
yeah,
I'm not
without
sure.
recombination, I think it is. Yeah, I don't know. So I don't have an answer to this question.
Okay. And another question, if indeed it's about pathogens and the immune response reducing the lifespan to protect basically the whole population, could you take some fruit flies and introduce some STIs and see if that changes the lifespan? Or is that just a too simplistic of an approach? Like, can you take a group of fruit flies, give it some STIs, and see if over generations it evolves to reduce its lifespan?
Yeah, we were thinking about these experiments. So I just want to specify that you said that for the population is a wrong take, right? So we are kin selection model, so it should be good to our kin. So and to take like a viscous to build a model of viscous population, probably with worms, and then try to model something like that with the virus. So, but I was considering that. We never did it. But when I'm trying to construct that in my head, I realized that there will be way too many like interventions that I need to apply, that the reviewer will say that I have constructed the result. So, and that's why I think they producing predictions about species in nature and trying to follow this up is a sort of a stronger approach. Also because many of these experiments are very kinematic, and this also something that people like. So we were discussing it with people like to build like viscous population models in C. elegans, but in C. elegans there are not many viruses available. So yeah, and flies are flying too much. So you need to have a very big compound to have a viscous population.
Thank you.
Peter, this was really thought-provoking. Thanks. I've got two questions. The first has to do with just wild populations of naked mole rats. Has the rate of parasitism and infection been looked at? Is it really zero in wild populations?
I think there are some bacterial infections. I'm not sure if they're infections or like commensal bacteria. So I think I don't remember any reports about virus in like mole rats.
And so they don't get intestinal parasites or anything like that?
I don't. Yeah, this I don't know. This I don't know.
Okay.
This is something to look at.
Yeah, that'd be really interesting given your hypothesis. And then my second question has to do with some of the lifespan extending interventions, such as like rapamycin or caloric restriction. How do you think those are tapping into this kind of immune response? Are these suppressing that, or are they enhancing it, or?
I think it's known that chronic infections or chronic inflammation can accelerate aging, right?
Right.
So it is true for HIV, let's say, according to the epigenetic clocks. We have found, the slide is not here, in Drosophila, also the virus infection can accelerate aging dramatically, according to the transcriptomic clocks. Also, another lab recently put out a preprint. You can look it up. I can drop you the reference if you want. So because our data is not yet out, not out yet. So, but idea is that organisms are modulating the speed of their aging. If they feel chronic infection, then they try to accelerate the human endpoint. And that's why, for example, anti-inflammatory peptides from worms are prolonging lifespan. And I think rapamycin is immune suppressor, right?
It is, that's right. Yeah.
And I think for metformin, it was shown that it's anti-inflammatory. Ozempic is anti-obesity, and obesity is also associated with inflammation. So I think they all fit into the inflammatory paradigm.
Thank you.
Yeah, I have two questions too. The first one you already more or less answered. It was about if finally to have a slightly weak immune system would help for lifespan. But you mentioned rapamycin. It was my question because it has this immunosuppressor effect. And my second question is, could it be possible that regeneration is also a pathogen control method, particularly for example, planaria? Because it removes like a lot of cells in one time when they reproduce by fission.
No, yes and no. So of course, if you can, let's say, drop the part of the body which is infected and then regenerate it, it can be a pathogen control. And I believe that skin, blood, and guts in humans are regenerated partially because of this, because these are like exposed units that are, no, blood maybe not, but guts and skin for sure, that they are exposed to pathogens, and it's better to just remove it and build new ones that the viruses that infected them are also removed. However, I think in planaria, regeneration doesn't happen routinely, so they don't shed cells out, right?
No, they do. Yeah, I mean the asexual forms not only do individual cells senesce and die, but every couple of weeks they'll fission and they'll drop tail and and fission and yeah.
No, I don't think this can help.
Yeah, I've heard, by the way, I think I remember this correctly. There's a snail that gets some sort of parasites in its head, right? And they they self-amputate the head or something.
No, other way around. The head, I think, is uninfected, so the
The
body
head
is
stays.
infected.
I see.
It chops the body. Yeah.
I see. Okay. Anybody else? Because I've got a couple of questions, but you guys go ahead if anyone else. Okay. Yeah, thanks, Peter. That was really interesting. So a few things. First of all, what are your thoughts on good markers? The relevant markers of aging, and other than waiting till the organism actually dies, of course. But are there molecular measurable molecular or physiological qualities that you think track the aging process?
Oh, this I cannot answer, I'm afraid. So I think whatever is connected to immune system is changing very dramatically and very early. So in at least in humans, it's like that. Also in flies, we see that the immune system changes. So in flies, maybe it's a little bit different because there is some of the molecules are making, some of the proteins are making like a stepwise increase. So, but I believe that aging is a immune thing, and I believe that, and also I think a recent publication showed that immunity has a huge contribution to the methylation clocks, right? So that biological age goes up if people are infected with the COVID, and goes down if when they recover. So, which is actually a bad marker, right, in the end. But if we speak about humans, I think some markers of inflammation would be probably most relevant according to my model. But I don't have like solid evidence or experiments in my hands to recommend anything.
Yeah, I was just wondering because in the field, like a lot of metrics somebody uses routinely, and then somebody else will say, this is sort of a side of this is not actually fundamental to aging, right? There's a lot of debates about what are the markers that you can use to see how your strategy is working. Another question: do you know? And I asked this of everybody, and I still don't have a good answer. What do axolotls actually die of? Do you have any idea?
Whom?
Axolotls. The highly regenerative amphibia. Yeah, I mean,
no, of course I don't know. I spoke to Maximino June, and this is of course a fantastic model, and it also challenges all the like damage accumulation because they don't have epigenetic aging. It seems to be.
Yeah.
So, but I'm also very excited about that. But I think I also don't know what they should die. So, but my model in principle accommodates it if they really develop an alternative like self-killing mechanism, then it fits very well. So then it means that the thing is not aging itself, but dying, the key. So then it fits my model very well.
Got it.